And so the next award is the Life Achievement Award, and so it's my great pleasure and honor to introduce this year's recipient for this Lifetime Achievement Award, Eugenia del Pino. So Eugenia was a pioneer in the field of Evo-Devo, as you will hear, and so I think the recognition of her work is particularly appropriate in the context of this joint meeting with the Pan-American Society for Evo-Devo. So Eugenia studied at the Pontificia Universidad Catolica de Ecuador, also known as Pourquié, I guess, in Quito. She left to do her PhD in the US at Emory University with Asa Alan Humphreys, working on the fertilization of xenopus eggs. And Humphreys trained with Fankhauser in Princeton, who trained with Balzer, who trained with Theodore Boveri. So you can trace back the lineage of Eugenia all the way up to Theodore Boveri, who, as you know, discovered the role of chromosomes in heredity. So after she graduated, she returned to Pourquié in Quito, where she became professor of biology. And that's where she spent most of her career, with some shorter stay in Germany and in the US. And so, because she had very limited support there for her research, she could not continue to work on xenopus that she had been working on during her PhD, because she could not afford the cost of the husbandry. And so she decided to turn her interest to local frogs that were found on the campus of the university. There she realized that one of the species of frogs called Gastrotheca riobambae presented very unique developmental features that were not understood. So these frogs are called marsupial frogs, and they're very peculiar because they carry their embryos on their back in a pouch, and the development of the embryos is a very specific features, some of which remind of the development of vertebrates and the placenta. And so she studied this frog for most of her career, and was interested in two aspects, which I suppose she would discuss. One is the multinuclearity of oocytes, so an observation that she made that some of the oocytes, some of the species of these frogs have multinuclear oocytes, whereas in xenopus, for instance, and most of the frogs, there is only one single germinal vesicle. And also she made important contributions to the study of gastrulation, showing that in this species, gastrulation proceeds in a fairly different manner compared to xenopus. And so the embryos forms as a disc, pretty much like a bit more like in chicken embryos, and there is a dissociation between gastrulation and elongation of the embryo compared to xenopus. So she published nearly 100 scientific papers, and her important work was recognized by many accolades. So she was elected to the Latin American Academy of Sciences and to the World Academy of Sciences for the advancement of sciences in developing countries. And importantly, she was the first scientist from Ecuador to be elected to the American Academy of Arts and Science and to the National Academy of Sciences of the US. She received several prestigious prizes, like the Eugenio Espejo Prize, which is the highest award given by the government of Ecuador. And she also received the 2000 L'Oréal UNESCO Prize for Women in Science and the 2019 Prize in Developmental Biology of the Latin American Society for Developmental Biology. And so apart from her lab work, Eugenia was also very much involved in the politics of science in Ecuador, and she was a founder of the National Academy of Sciences of Ecuador, which is now thriving with more than 50 fellows. And she also devoted a lot of energy to the conservation of the Galapagos Islands, where she served as a vice president for the for Ecuador of the Charles Darwin Foundation. So just to conclude, I think overall, Eugenia's career perfectly exemplifies that what matters most in our field is not so much the resources, but the passion for research. And so please join me in congratulating Eugenia.
I thank the SDB for this prize. My talk will be divided in 3 parts. First, I will explain about the importance of the SDB in my career, and then I will explain about aspects of oogenesis and gastrulation, particularly in the marsupial frog from Quito. The scientific name of this frog is Gastrotheca riobambe. It belongs into the family Hemiphractidae. Excuse me, I don't know how to exchange this. The SDB promotes scientific careers in young people, and I think that my career exemplifies this aspect. I attended my first scientific meeting of the SDB and my first scientific meeting at the Southeastern Developmental Biology Conference in 1971 in Tallahassee, where I presented the work of my PhD in progress. In the information for authors, it reads, as it has been the case from previous conferences, one of the chief aims will be to give graduate students and other young investigators an opportunity to present their work. After this meeting, I became a member of the SDB, and I kept this membership for many years. Each year, I received the bound volume of the scientific conferences and also some leaflets concerning the teaching of developmental biology. The scientific resources that I had were limited, and therefore this was a very important source of information for me and for my teaching. At Emory University, my advisor was Dr. Alan Humphreys. He studied his PhD at Princeton University and was a student of Gerhard von Hauser. He had a very important academic genealogy that goes back to Theodore Boveri. One of the students of Boveri was Hans Spemann. Another one was Fritz Walser. Walser was later a professor in Switzerland at the University of Bern. His first graduate student was Gerhard von Hauser. Von Hauser applied to a fellowship from the Rockefeller Foundation, and when he finished his PhD, he came to the United States. Towards the end of his stay, he visited Professor Conkling at Princeton, and Conkling suggested that he applied for a position, as he did not have a position for him waiting in Switzerland. He applied and he became a professor at Princeton. And in this way, thanks to the Rockefeller Foundation, a direct line of scientific descent from Theodore Boveri came to this continent. Humphreys spoke very highly about the Boveris, Theodore Boveri and his wife, Marcella O'Grady. He used to say that it was actually Boveri who made it understood the mechanism of cell division. Boveri discovered meiosis. He and his wife discovered the interaction of the cytoplasm and the nucleus for embryonic development, and the importance of the centrioles for the division cycle. Marcella O'Grady, she was a professor at Vassar College, the director of the biology department, and she went to Boveri's laboratory for a sabbatical. They got married, and this meant that she never returned to Vassar College. Years after his death, she returned to the United States, and among her belongings, she had some slides, microscopic slides, prepared by Boveri himself. Years later, Joe Gall had access to some of those slides and took this beautiful picture of the centrioles in an egg of ascaris, a preparation done by Boveri himself. I have to say that I am extremely fortunate to have this very important scientific genealogy. But I am even more fortunate because in my lifetime, I have received the support and the encouragement from many scientists, and I am grateful. And I think this is a special opportunity to express my gratitude. Let me tell you about my PhD thesis. It has to do with the fertilization process in Xenopus, in particularly in regard to the frog egg jelly. I will only explain about an observation and an experiment. It was known that the egg jelly consisted of 3 layers. So one day, I took a dish of low salt solution, and I placed an egg in the solution and observed it under the microscope. And I was so happy because I could see in the living egg the 3 layers of egg jelly. I left and went to do something else and came back, and to my surprise, the morphology of the egg jelly had changed tremendously. I tried to fertilize this egg, and the egg was no longer fertilizable. I then did an experiment. I took an egg of Xenopus and placed it in distilled water for 1 hour. And I checked the condition of the jelly, and it had a non-fertilizable condition. And then I moved into a dish of physiological saline solution. And you don't imagine, it was immediate. The jelly changed to the 3-layer condition, and of course, I was able to fertilize this egg. I graduated in June of 1972, and the following month, I returned to Ecuador. I was very lucky because I was able to find a job at the Pontifical Catholic University of Ecuador. It's important to have a job. The problem was that I had no research funds, and so I could not work with Xenopus anymore. And my idea was, well, I will go to the gardens of the university, find a frog, and continue with the studies of the frog egg jelly. I never returned to the frog egg jelly studies. I found this frog, the marsupial frog. It was actually a female with embryos. This frog carries the embryos in the back as if it were in a backpack. I wrote to my advisor, Alan Humphreys, and I said, please go to the library, go to the Emory Library, and please give me the information about all of what is known of the development of this frog. He wrote back and said that nothing was known about this frog, the development of this frog. that I had an open field in front of me, and that if I wanted, I could do my research by analyzing the development of this frog. I followed his advice, and in this way I satisfied my intellectual curiosity, and I had projects for my students. Let me come to the study of oogenesis. And you are probably thinking, why oogenesis? I want to give you the background. One of the themes of the Humphreys laboratory was actually oogenesis. And he went in a sabbatical to the laboratory of Herbert McGregor in the UK. When he returned to Emory, he gave a seminar where he mentioned that inside the oocytes of the North American tailed frog, there was not one nucleus, but 8 nuclei. The nucleus of the oocyte is called the germinal vesicle. I thought that this was fascinating, and I thought at that time that the North American tailed frog should not be the only frog that has this kind of oogenesis. Therefore, back in Quito, I looked at the oocytes of all of the available frogs. On the whole, there were 51 species. The majority of them had a single nucleus in each oocyte, and I call this type of oogenesis the mononucleated type of oogenesis. In other species, there were from a few nuclei to thousands of nuclei in each oocyte. These are I call them multinucleated oocytes. Multinucleated oocytes were found in 12 species. All of them belong into the family Hemiphractidae. Unfortunately, the marsupial frog from Quito has mononucleated oocytes. I checked carefully, and we did not find a multinucleated phase during oogenesis of this frog. Other species of Gastrotheca, larger frogs, produce the largest frog eggs that are known, of 1 centimeter in diameter. In the ovaries of such frogs, the oocytes have what you see, these round structures, those are nuclei. These are multinucleated oocytes. I studied multinucleated oogenesis in greater detail in the frog from Venezuela, Flectonotus pygmaeus. I opened up an oocyte of Flectonotus in a dish of saline solution, and it was incredible. There were thousands of these round spherical structures. I wrote my observations to my advisor, Alan Humphreys, and he was so surprised that he traveled to Quito to see these extraordinary oocytes for himself. With his help, we detected that, in fact, these were nuclei surrounded by a nuclear envelope. Inside each one of these nuclei, there were nucleoli. The nuclei were active in RNA synthesis. In the drawing, I explained. At first, all of the nuclei were of about the same size. As the oocyte grew, the nuclei of the central part remained small. The peripheral nuclei enlarged and also developed Lambrach chromosomes. And in the final oocyte, there was a single nucleus. I took a few specimens of Flectonotus to the laboratory of Herbert McGregor in the UK to analyze for the amplification of the ribosomal genes. We detected that each nucleus of a Flectonotus oocyte is a meiotic nucleus with 4 times the Z value for this species. We detected that each nucleus amplified the ribosomal DNA. The level of amplification varied between nuclei, and the level was very low. However, when one considers that each oocyte of this frog had between 2,000 to 3,000 nuclei, the total level of ribosomal gene amplification was far higher than in Xenopus. I couldn't continue with the analysis of multinucleated oogenesis because I could not collect frogs. They are very rare, and so due to the lack of material, this project was discontinued. But I want to give you my interpretation of the multinucleated oogenesis. Nerve cells have been found in animal oogenesis in all of the animals that had been studied. Well, an example is the Drosophila. The mouse has nerve cells. I consider that the multiple nuclei in oocytes of marsupial frogs are nuclei of nerve cells that are placed in the cytoplasm of the oocyte. My interpretation is that the evolution of terrestrial reproduction invertebrates required large oocytes, and to make a large oocyte, the nerve cells, or in this case, the multinucleated condition, is required to make the oocytes. I now will come to the part of gastrulation. But before, let me tell you about the eggs and the culture of embryos. The marsupial frog is a frog, and I have to report that, in fact, the oocyte has egg jelly. That's wonderful. I could not determine the conditions for the in vitro fertilization of this frog because I could not keep in culture the eggs and sperm. Embryos die in low salt solutions. Embryos die in physiological saline solutions. I was able to culture the embryos when I added urea to a physiological saline solution. Urea acts as an osmolyte for the preservation of water in animals that live under conditions of water stress. And so here is a picture of a fixed embryo of the marsupial frog seen from the vegetal hemisphere, and you can see that there is a blastopore around a yolk plug. As in Xenopus, there is involution occurring at the blastopore lips. The difference is that the cells that involute remain in the blastopore lips. They will form an embryonic disc, whereas in Xenopus, the cells that involute move away from the blastopore. If at closure of the blastopore, if the embryo would have been stained for cell borders, there is an area of small cells surrounding the closed blastopore. The body of the embryo develops from this embryonic disc. This is the embryonic disc. So this resembles the condition in the chick, but the chick does not gastrulate through a blastopore, so it is a different process. And these frogs have holoblastic cleavage. It was of interest to see whether other frogs may produce an embryonic disc at gastrulation. And we analyzed 16 species that represent 6 reproductive modes. Frogs with aquatic dependence for reproduction, for example, in Xenopus that have aquatic eggs. Frogs that deposit the eggs in foam nests that float in the water. Frogs that deposit the eggs in the leaves of plants surrounding the water, and when hatching, they fell into the water. Terrestrial eggs occur in the poison arrow frogs, the dendrobatids. They deposit the eggs in the soil. At hatching, the tadpoles are transported by the parents to the water. Eleutherodactylus is the frog that does not have a tadpole. And the marsupial frog, of course, incubates the embryos in the body of the mother. Excising these frogs, in this image, you can see that the frogs with terrestrial legs have a tendency towards larger eggs. The analysis of gastrulation revealed differences in regard to the elongation of the notochord, and I called two modes of gastrulation. Gastrulation mode 1, in which elongation of the notochord begins in the midgastrula, and gastrulation mode 2, in which the blastopore closes and only then the notochord begins its elongation. Gastrulation mode 1 is the mode of gastrulation in Xenopus. In Xenopus, blastopore closure is guided by two processes, convergent thickening and convergent extension. And a gastrula of Xenopus stained for brachyury shows that there is a yolk plaque and the notochord is already extending. This is the gastrulation mode 1. This kind of gastrulation was found in frogs with small eggs and aquatic reproduction, that is, the ones with aquatic eggs, with home nests and arboreal eggs. Gastrulation mode 2, these are embryos of dendrobatid frogs, and you can see that the blastopore is there, and there is no, the yolk plaque is there, and there is no elongation of the notochord. Once the blastopore is closed, the notochord elongates. This shows demonstrates the modular nature of gastrulation. Frogs with this type of gastrulation are those with terrestrial reproduction, the dendrobatids, the frog without tadpoles, and the marsupial frog. I checked to see whether the frogs that delay notochord elongation, whether they produce an embryonic disc. No, they don't produce an embryonic disc. The cells that will form the notochord are at the closed blastopore lip, and the archenteron enlarges during gastrulation, whereas in the marsupial frog, the archenteron enlarges only later. In this talk, I had explained that the marsupial frogs exhibit a tendency towards larger eggs. I explained that they have the mono and multinucleated modes of oogenesis, that these frogs gastrulate through a blastopore like other frogs. I explained that they show the gastrulation mode 2 with retardation of the extension of the notochord. They develop from an embryonic disc. I interpret that these changes allowed the conquest of terrestrial life with parallelism with the amniotes. I wish to thank my former students, to thank my international collaborators, and to thank the Pontifical Catholic University of Ecuador for its support and for giving me the academic freedom to conduct my work. I thank the SDB for awarding me the Lifetime Achievement Award. I thank you so much for your attention.